Welcome back from the break. We're on the home stretch now, right into our next presentation on Q2R2, Q14, revision of Q2R1, analytical validation and analytical procedure development by Dr. David Keire, Director, Office of Testing Research in the Office of Pharmaceutical Quality, CDER, FDA. Next, we'll learn about Q5AR2, viral safety evaluation of biotechnology products derived from cell lines of human or animal origin by Dr. Chris Storbeck, Senior Quality Evaluator at the Cell, Gene Therapies, and Radiopharmaceuticals Division, Center for Oncology, Radiopharmaceuticals and Research Evaluation, Health Canada. And our final presentation in this session is on Q9R1, Quality Risk Management by Stephen Mahoney, Head of Quality and Quality Policy and Advocacy, Gilead. Please join me to welcome our first presenter, Dr. Keire.
Hello, my name is David Keire. I am going to be presenting on the subject of two ICH guidelines, Q2R2 and Q14. Q2R2 is a revision of Q2R1, which is a guideline on analytical procedure validation, and Q14 is a guideline on analytical procedure development. Before I begin, I want to just say that everyone deserves confidence in their next dose of medicine, and that pharmaceutical quality assures the availability, safety, and efficacy of every dose. Pharmaceutical quality is the focus of the office that I work in at the FDA. And what do we want? We want fewer drug failures. We want to minimize and eliminate drug recalls. We want regulatory flexibility and fewer filings. We want to lower barriers for new analytical technology and ultimately quality drugs for consumers. Now, I don't want to get into a discussion about what is the central science, whether it's physics or chemistry or biology. I'm sure many of us have opinions in that. But if you think about steps in a drug's life cycle, from drug development to manufacturing process monitoring and control, to quality control and batch release, and ultimately post-approval surveillance, all of these steps have key decisions that are made based on measurements. Analytical procedure measurements are providing the data that drive decision-making across all this landscape. So it behooves us to have the most reliable and robust measurements that we can get, and that's really the focus of Q2, R2, and Q14. So how do these two guidelines help? They standardize. They provide guidance on the content of sections of the common technical document. They harmonize, depending on how you spell it, and so that there's common requirements globally. And ultimately, together, they provide a framework that's adaptable to technological change and allows for continuous improvement for analytical procedures. So Q2R1 was finalized in the 90s, and a lot of scientific and technological progress has been made since the document was written. There's a lot of advanced therapies that are in drug development and commercialization, and the associated analytical techniques used to make measurements on those therapies are multiplying. There's a lot more use of hyphenated techniques, things like liquid chromatography, mass spectrometry, or spectroscopic approaches that are multivariate. For example, near-infrared or Raman spectroscopy tools. So for Q2, the existing version of Q2, what are the issues and the cost? As I mentioned in the previous slide, Q2R1 is not directly applicable to multivariate spectroscopy data. So with the lack of clear guidance, that leads to inadequate validation data and regulatory submissions, and ultimately to recursive information requests and responses that take time. And things like near-infrared technology is commonly being used for innovative techniques for example, things like real-time release testing. And so with the current version of Q2R1, there's kind of a barrier to innovation in analytical approaches for pharmaceutical quality assessment, which is why one of the reasons why it was decided to work on a revision. So for Q14, there has been no ICH guideline on analytical procedure development. And because of that, applicants rarely present performance evaluations, which they might do as part of an analytical procedure development in regulatory submissions. And that can lead to recursive regulatory communication around non-conventional analytical procedures. For example, things like process analytical technology-driven multivariate models used for process control. And the lack of a guideline and a place for these kinds of data in application impedes applicants from presenting a scientific basis, for example, quality by design data that might allow regulatory flexibility in post-approval analytical procedure changes. And ultimately, this delays access to medication and increases costs. So what is in these guidelines? What is developed is validated, right? So above this dotted blue line that's in the slide here lies Q14, where you use analytical procedure knowledge, can combine with what objectives you have to decide what performance characteristics you want, and you have analytical procedure related information from development. And based on all that information and knowledge, you move into Q2, where you write a validation protocol, you perform experiments, and perform evaluation of data, which you would write up in a validation report. And that provides a snapshot of the analytical procedure performance, and it's kind of a decision point that this procedure is going to work to measure the drug attribute that you want to measure. And after that, it moves into analytical procedure life cycle management, which crosses back into Q14. So in the previous slide, the Q14 looked like kind of a small component compared to Q2. But really, when you look across the analytical procedure life cycle, the tan box here is the Q2 part, and all the rest of the things are Q14 elements. So, if you look on the far left of the slide, where you have a drug product where you need to make measurements on, and you have some product and process understanding, and you know that you have some attributes that require testing, you develop an analytical target and you select technology and perform a risk assessment, identify parameter set points and or ranges, define an analytical procedure control strategy, and then you do the validation. After the validation, you go on to routine use and ongoing monitoring. You may have to do some changes to the procedure over time, because the validation is just a snapshot of the performance, and that can change for a number of reasons. I mean, your product may change, other things may change. You may want to, adopt a new technology. And so this slide basically describes what's in this box is all of Q2 and Q14. So I want to take a moment to talk about, in a little more detail, a couple of the things that I touched on in the previous slide. One of those things is analytical target profile. Now, the analytical target profile is, you have an intended purpose of a measurement, and you have some sort of performance target, and that can be described in this ATP. And that's really a technologically independent profile. You could have that performance. You can meet that performance target and intended purpose by more than one technology, more than one analytical procedure. And so based on your knowledge and a risk assessment that you perform that says, here's the most likely technology that's going to give me the performance I want, there's some risk associated with that. And then you make a technology decision, which leads to an analytical procedure attribute that you can measure. And there's associated criteria with that. And of course, all the analytical procedure parameters that go into that need to be considered and are a part of the equation of analytical procedure development as described in Q14. Now, another element I want to touch on briefly is when you need to make a change to an analytical procedure, you have to do a risk assessment there as well. And the risk of that change would depend on the extent of the change to the procedure, which critical quality attributes you're measuring, how important it is, how complex your product is your matrix going to interfere? And hopefully, you can control those risks by, what you know about the product, what you know about how the product was made, the process knowledge, and also about what you know about the analytical procedure, which you might have developed or might have accumulated during development of that procedure. And if the control of those risks outweigh the risks, you can decide to make that change. In addition, you could provide all these kinds of data and analysis of the data, assessment of those data, to a regulator to reach agreement about a reporting category using Q12 tools, which give you some post-approval regulatory flexibility. Now, I won't be able to go into all the other wonderful things that are in Q14 and Q2R2, but I've touched on a few of them, and hopefully I've stimulated enough interest that you will feel the need to go and read the entire documents. And they're available to you because we reached step 4 in November of 2023 after 5 years of work by the expert working group. And we're currently working on training materials as a IWG right now that will hopefully become available in about a year's time. I think in summary, I want to say that together ICH Q14 and Q2R2 describe the development and validation of analytical procedures used for the assessment of drug substance and drug product quality. Q14 describes the scientific principles for development, change management, and submission of analytical procedures using minimal or enhanced approaches. Now, I didn't really provide much touch on minimal versus enhanced approaches, but again, it's available to you in the document, and there's training materials to come. And ICH Q2R2 provides guidance for establishing and submitting evidence that an analytical procedure is fit for assuring drug quality, just like R1, but it also allows, it has more flexibility for innovative technologies or new or different technologies besides HPLC, and hopefully it's an adaptable framework that can be applied to any analytical approach that you might take. I also want to say that Q2 and Q14 exist within a continuum of many different ICHQ documents, quality documents, and we pulled from many of them in the process of putting together the manuscripts. And I just point out a few of them here. Q2, Q8, R2 on pharmaceutical development, Q9 and quality risk management, and Q10 pharmaceutical quality systems, Q11 development and manufacture of drug substances, Q12 on pharmaceutical life cycle, and Q13 on continuous manufacturing. All of these things were considered and when we developed our guideline, and they kind of all work together for a common goal of assuring drug quality. Now, this picture was taken in Prague in November of 2023, and you can see how happy everybody is because we've actually just, at this point, we had just signed off on those two documents after
Good afternoon. It's a pleasure to be here this afternoon to discuss the revision to ICH Q5A, viral safety of biotechnology products derived from cell lines of human or animal origin. I'll begin with a brief outline of the presentation. So I'll start with some background on the revision. I'll then transition into key principles of the revision and then talk about the key updates that were incorporated into the document. And I'll finish with some conclusions. In terms of background, ICH Q5A was introduced in 1999, and for the past 20 or so years, it's been a very useful document. However, it was recognized that in light of scientific advances and advancing technologies, a revision was in order, and so a concept paper and business plan was introduced in November of 2019. And for the last 4 years, the document has evolved and has been changed, with the Step 4 document being released last November in Prague. So I'll go over some of the key principles of the revised document. And these include the overall retention of the basic organization and the scientific principles of the document based on the utility of the original. There was a recognition that the advances in science and technology did necessitate an update to the document. And these scientific advances that have been written into the new revision allow for flexibility for continued scientific advancement. The EWG was careful indeed to not paint themselves into a corner. The new technologies introduced into the document align with the 3R principles of replacement, reduction, and refinement of animal testing. And there's been an introduction of advances in viral safety as well. Certainly the application of new technologies such as next generation sequencing has been incorporated into the document. The introduction of platform approaches to viral clearance. New product types have been incorporated, as well as viral safety considerations for continuous manufacturing. So the objective of the document remains consistent with the key scientific and regulatory considerations with respect to virus clearance, characterization, and testing. So the three principal complementary approaches to virus safety remain intact, which are the selection and testing of cell lines and raw materials for virus contamination, assessing virus clearance capacity of the process unit operations, and also testing for infectious viruses within the product at the appropriate stages of manufacture where they're most likely to be detected. Q5A R2 is designed to be used in conjunction with other ICH guidances, namely ICH Q2, Q5D, and ICH Q13. Public consultation took place last year, and a number of the comments were incorporated into the document. A lot of them concern the new products, so clarifications regarding the new products. Some new definitions were required to take into account the integration of new concepts into the document. Clarification in terms of some principles such as in vitro cell age and end of production cells, which is a terminology that was introduced with the new products. Also implementation of next generation sequencing and some more clarity in terms of how that is being integrated into the document. Additionally, there was a removal of Annex 1, and this annex dealt with the production of product in vivo, namely ascites fluid, and this was removed. Pardon me. In terms of the table of contents, the introduction was subject to some significant changes with the introduction of new product types and new concepts of viral clearance and viral safety. Sources of viral contamination was only subject to minor changes. Sections 3 through 6, major changes took place with these sections in terms of cell line qualification, testing for viruses, viral clearance studies, and evaluation of viral clearance. A new section on continuous manufacturing was incorporated, and as I mentioned, the glossary was changed significantly to reflect some of the new concepts and modalities introduced into the document. In terms of the annexes, the first, with the exception of Annex 1 from the original document, which was removed, the first 4 annexes in the new document remain somewhat unchanged. 2 new annexes have been introduced. One detailing examples of prior knowledge with respect to virus inactivation and clearance, and a new annex detailing aspects of genetically engineered viral vectors and the testing and viral clearance that is required for those new product types. In terms of the new product types, key update 1, the scope was expanded to include viral vectors. And these viral vectors are vectors that are amenable to viral clearance. So for example, non-enveloped viruses, which can withstand some of the virus inactivation conditions. Genetically engineered viral vectors and viral vector-derived products. So these are products that are where viruses are used in their production, and they are not actually the products themselves, the viral vectors. So these can be used to produce recombinant proteins, such as in the baculoviral system. Additionally, viral vectors, where viral helper virus may not be required for their production, have been incorporated. So, for example, the triple transfection of cell lines for production of AAV is a good example. In terms of viral vector-derived products, an example of this could be proteins manufactured from viral vectors for generation of virus-like particles, and also the protein component for nanoparticle-based vaccines with the protein product as the delivered product. So the overall sections were altered to incorporate this new information for new product types. For example, there was additional reference in Section 2 for sources of potential virus contamination being updated to reflect for these new product types there will be virus seed banks. Additionally, in Section 5, which details the rationale and action plan for virus clearance, a new case F was introduced regarding the clearance of production or helper virus. So in terms of the use of the relevant model virus for clearance. A good example, so the examples of viruses utilized in clearance studies were updated to include baculovirus, for example, as one such virus type that could be used for the production virus. And in terms of Annex 6 and new product types, this is a new annex detailing specifically virus safety considerations for these new product types, including information on testing in a new table where these expectations for testing for these products are detailed. Key update 3 is continuous manufacturing. So there's an entirely new section dedicated to virus safety and continuous manufacturing. And this deals with the specific aspects of continuous manufacturing that have to be considered in terms of virus safety. And it's designed to be used in parallel with the ICH Q13 guideline on continuous manufacturing. And as I mentioned, specific aspects of continuous manufacturing and how these are dealt with respect to virus safety are outlined. So for example, with cell cultivation, the length of time that the cells are being cultivated in the continuous process has to be considered. in the event of a contamination event, how the material is treated or separated, if that's if such an occasion arises, and the thought process behind that was integrated, as well as the integration of unit operations. So how these unit operations are thought of. Are they thought of as batch operations as a starting point for dealing with the virus clearance considerations, with the virus safety considerations? Additionally, so in terms of specific considerations for unit operations, such as the chromatography, if there are the chromatography columns used in tandem, in terms of low pH and solvent, what is the residence time and the exposure time of the product to the inactivation agent, the other considerations that are specific for those inactivation steps. In terms of viral filtration, considerations of the product flow rates through the virus filters, the interruptions in terms of the filtration process. All these elements of filtration have to be considered with respect to the ability of the filter to clear the viruses effectively. In terms of new test methods, the main new technology being introduced into the document, and it impacts a large number of sections of the document, is the introduction of next generation sequencing. Excuse me. There is a new section in the cell line characterization section of the document detailing molecular methods, next generation sequencing being one of them. In terms of the application of the technology, which could be used to replace the in vivo test or supplement or replace the in vitro test, it's stated clearly in the document that head-to-head comparisons are not recommended or expected. NGS is considered a limit test, and so it must be demonstrated capable of detecting low levels of a contaminant, and as such should be appropriately validated for that use. In terms of key update 4, the NGS or the nucleic acid amplification techniques may also be used as an alternative to certain virus-specific detection methods, such as the MAP or HAP or RAP antibody production tests. And that, again, is in line with the concept of the 3 Rs. So recommendations regarding the uses of NGS or nucleic acid testing are described throughout the document. Key update 5 is resin reuse, and there's been certainly a large amount of scientific progress in the last 20 years with respect to understanding of the resins being used. For example, in the protein A columns, the affinity capture chromatography, it's understood clearly now that virus removal is not impacted or actually slightly increases for used resin. And as such, the guideline is clear in stating that the product-specific resin reuse is not an expectation. As for other resin types, the guideline remains open-ended regarding the use of prior knowledge. For example, the anion exchange chromatography or the cation exchange chromatography. And so for these resins, or perhaps it's a little less clear, but there may be some advances in this regard. Equivalent prior knowledge should be provided in place of product-specific viral clearance studies. So key update 6 deals with prior knowledge. So a new section has been added in section 6 containing specific principles required for the use of prior knowledge. So, for example, a well-characterized platform process must be used. So, speaking to a large amount of experience with the particular process. The composition of process and intermediates. What are the impurity levels? They should be similar with the similar process. In addition, the order of unit operations should be the same, with an equivalence of the upstream step to the one being validated for viral clearance. And the robustness of the critical parameters should be demonstrated as well. In line with this, a new annex has been created, Annex 5, that outlines 3 virus inactivation or clearance steps and the process parameters that have been characterized in terms of their effectiveness for virus clearance or inactivation. And they are regarding the low pH detergent solvent inactivation and also the virus clearance with the virus filter. Key update number 7, the flexible approach for well-characterized substrates, details testing flexibilities for cell substrates that have a lot of prior knowledge, such as CHO, for example, which has a long history of safe use, and is also the retroviral particles have been demonstrated to be non-infectious. Annex 4 includes a footnote in the safety factor calculation where the safety margin is indicated that a margin of less than 10 to the minus 4 particles per dose may be acceptable for these particular cell lines. Additionally, the use of CHO-derived endogenous virus particles may be used in virus clearance studies, with the caveat that the molecular or biochemical detection method be qualified and demonstrated to be fit for purpose. In addition, in vivo testing may be excluded in certain situations. For example, where a parental cell line has been used to manufacture or produce a number of master cell banks, and also the controls for the generation of master cell banks from that parental cell line are well characterized. So it's clearly stated in the document that in vivo testing is not necessary for extensively used, well-characterized cell lines such as CHO, NSO, sp20, based on prior knowledge. And the glossary has been changed, of course, to reflect new definitions for new product types and new concepts that we just spoke of. For example, next generation sequencing, and definitions regarding expectations for new products, such as the helper virus, viral vector for protein expression, viral vector derived products, and master virus seed and working virus seed and production virus. All concepts that need to be clearly defined. Additionally, the new concept of prior knowledge had to be appropriately defined, including the definitions of platform validation and platform manufacturing, process robustness of virus clearance, what is prior knowledge exactly. Additionally, as I touched on earlier, definitions to align terminology that has been introduced due to new product types such as end of production cells, and the legacy definition of in place for the limit of in vitro cell age. Those had to be aligned, as well as jurisdictional differences in terms of defining things slightly differently, such as extended cell bank. And these had to be harmonized in the document. In terms of implementation, it was stated in the document that products not specifically indicated in the scope, the concepts of the document can still be applied and be useful. In terms of validation of new technology, such as next generation sequencing, the guideline has not been prescriptive in how to validate, and it's encouraged that the users of the document read the revision in concert with ICH Q2 and also ICH Q5D and ICH Q13. So in conclusion, the ICH Q5A revision establishes harmonized scientific and technical requirements for products derived from characterized cell lines of human or animal origin. And it helps outline regulatory expectations for testing, evaluation of virus safety. The guideline has retained the original structure and principles of the original guideline and provides additional recommendations on the 3 established approaches to control viral contamination, including the selection and testing of cell lines and raw materials, the assessment of virus clearance capacity of the processes and unit operations, and of course the testing of the product at the appropriate stage of manufacture where it's most likely to be detected. So with that, I'd like to thank you for your attention, and I'd be happy to take any questions.
Good afternoon, everyone, and welcome to this presentation on the topic of quality risk management. I'm Stephen Mahoney from Gilead Sciences, and I currently am the Pharma Topic Lead for ICH Q9R1. So on behalf of Pharma, I wanted to thank the FDA and Health Canada for the opportunity to present on this important topic of quality risk management. Here's a short disclaimer, understanding that nearly all the information contained in this presentation has been adopted or slightly modified from several publicly available ICH presentations. I also provided a link to this information at the end of this presentation. Moving on, here's an overview of the information I'll be addressing over approximately the next 15 minutes. I will start by describing the general purpose for the revised ICH Q9 guideline, and then highlight a few of the ICH process milestones for this Q9 revision. And finally, I'll delve into the real content of this deck, meaning a discussion of the revision topics themselves. So let's begin with the initial question. What was the purpose of the revision? Well, I think it's important to understand that the revision was intentionally designed to be a very targeted revision of the original Q9 guidance document, and therefore it only addresses specific areas for improvement. One of the areas is the concept of subjectivity, as there was a feeling that high levels of subjectivity in risk assessments and in QRM outputs have been seen in the industry and across the industry, and something that needs improvement. The second concept, of course, is product availability. It's something that's been in the news for many years, and it's something that obviously is a great concern to industry and to our patients. The topic of formality. Similar to subjectivity, I believe there's a lack of understanding as to what constitutes formality in QRM work. Similarly, for risk-based decision-making, it's a question of what is this and how is it used in the context of QRM. In addition to the development of new content for the above topics, there's one specific terminology changed in the original Q9 document. This is the term risk identification, which is modified to become hazard identification, which is more consistent with the original definitions. And for the sixth topic, risk review, there were concerns about a lack of clarity on the concept itself. As you'll see here, this is an important note. New guidance or training has been developed for each of these areas. The first 5 topics were addressed with new guidance in the ICH Q9 document itself and by developing training materials that support the new guidance. In the 6th topic, risk review, while it did not have any new guidance written for it in the guideline, new training material was developed for this topic. But before we go into the specific areas, let's take a step back and look how we got to where we are today. Given our limited time, I won't go into too many details for these milestones, but I do think it's important to understand that the overall time frame for this targeted revision took several years of hard work and dedication by many individuals from many different organizations around the world. I also want to quickly note that I provided a link here to the ICH concept paper and the business plan, as well as a final guidance document for May of 2023. And then in terms of process, I also wanted to note that the EWG reviewed and adjudicated over 750 comments received during the public consultation period, which started in 2021. As you can imagine, this was a very time-consuming process, but in the end, the expert working group was able to reach consensus on all the final changes. And as you can see here on the bottom, the IWG now is currently working on supportive training material with an eye to complete this work in 2024. So here are the revision topics we identified earlier. Let's go one by one with some details and descriptions. So topic 1, Subjectivity. Probably the first question I assume in everyone's mind is, what is subjectivity? Well, as I was preparing for this presentation, I looked at several dictionary definitions. I remember doing this a few years ago when the EWG began, and I found a few different ideas that I thought essentially captured what we were thinking about. But essentially, subjectivity is something that's based on an individual's perceptions, feelings, or intentions. So here's a picture from one of the past EWG presentations. When I see this, I see a few things. One person's looking at the object on the left as a tree. Another person saw this same thing as something that produces an apple. And the person on the right saw this as something that can generate money. So I think you get the idea. It's fairly easy for three people to look at the same thing and leave with three very different perceptions. Given this, let's move to a little more detail on the topic of subjectivity in terms of Q9. I think we can all agree that high levels of subjectivity in risk assessments in QRM are problematic. And why is this? I think it's pretty obvious. Subjectivity may directly impact the effectiveness of the risk management activities and the decisions made. Well, again, I don't think this idea is particularly novel to anyone listening to this presentation today. So I think the more important question really is, what causes subjectivity? Again, of course, this isn't a simple answer, and there's many, many factors. The ICH Q9 guideline addresses and discusses several of these, including differences in how hazards, risks, and harms are perceived by different stakeholders. This, of course, captures the ideas of biases. Another issue we've discussed extensively with the expert working group is inadequately defined risk questions, which really starts the process off in the wrong direction. And three, another example is the risk scoring methods that some risk assessments use. Of course, given all these different risk tools and all these different causes of subjectivity, is it really possible to eliminate subjectivity? Well, again, I think everyone knows the answer is no. However, while subjectivity can't be eliminated from QRM activities, it may be controlled using well-recognized strategies, including addressing biases I mentioned earlier and other behavioral factors. I believe the specific language used in the Q9 guidance is that subjectivity should be, quote, managed and minimized. It's an important concept. Before continuing, I want to highlight the importance of the information in this yellow box. Given the nature of the targeted changes I spoke about, and the somewhat use of vague terms like subjectivity, I think it's really important to read the revised guideline and the ICH new training materials together. The training materials were developed essentially in parallel or soon after the revisions were made, and they really add a lot of substance to a lot of terms that within the guideline, again, are fairly vague. So the second topic we addressed in the revised ICH Q9 guideline involves product availability, otherwise known as drug shortages to many of us. But before I go into too much detail, I wanted to flag for everyone that the original guideline already addressed product availability risks through its definition of harm. The definition of harm was, quote, included damage from a loss of product quality or availability. So the idea and the concept is already there. Why did the concept paper and the business plan seek to increase the emphasis? Well, although we recognize ICH Q9 is not a supply chain guideline, it was agreed to develop and expand on this topic for several reasons, the largest of which is probably the idea of globalization. Therefore, addressing product availability risks more clearly and with more focus was important to the group, given the extent of globalization of medicine supply chains, the associated complexities, and the different types of fragmentation involved with this. Again, if you look in the yellow box, this increased focus on this topic is really patient-focused, meaning a better understanding of how to develop appropriate risk-based drug shortage mitigation and preventions will serve the interests of our patients. So turning to the guideline itself, the revised guideline addresses several things. It says that quality manufacturing issues, including noncompliance with GMP, are a significant cause of product availability. Again, I don't think this is really in question, but I think everyone probably knows it's well addressed in literature over the last 10 to 15 years, including something I worked on with ISPE in their drug shortage survey, which was cited back in 2013 in FDA's strategic plan for preventing and mitigating drug shortages. This is just one of many efforts and works that have been done by different trade associations. But again, it's been a long-term effort, and it's something that we're hoping to improve with this language. In B, the revised guideline also describes how an effective pharmaceutical quality system drives both supply chain robustness and sustainable compliance. In particular, there's specific language in the guideline that describes how an effective PQS uses quality risk management, and quote, to provide an early warning system that supports effective oversight and response to evolving quality manufacturing risks from the pharmaceutical company or external partners. This concept of an early warning system was something, again, that was discussed in great detail by members of the expert working group, and consequently, you'll find that idea captured in the guideline itself as well as the training. And then for C, in terms of formality, the guidance describes that when risk-based shortage prevention and mitigation activities are performed, the level of formality may vary and should be commensurate with the level of risk associated with the loss of availability of the products. Again, quite vague, but also I think it's important to realize there's flexibility here in terms of the level of formality. And then in D, the revised guideline also addresses several specific quality manufacturing factors that may affect product availability. And the guideline goes into a great deal of detail here. On 1, manufacturing process variability and state of control. 2, manufacturing facilities and equipment, including, I believe, a case study involving older facilities and older equipment. And then 3, the oversight of outsourced activities and suppliers. And before I turn to the next topic, I want to reiterate what I said on the first one, meaning it's very important to read the guideline and the training material together. For this topic of product availability, the EWG created a great deal of content for the training, based in part on expert material provided by several external stakeholders. Topic number 3 was formality. The original guideline said, quote, the level of effort, formality, and documentation of the quality risk management process should be commensurate with the level of risk. Again, I think this sounds great and no one can argue with it, but it begs the question, well, what does formality in QRM actually mean? I think it's safe to say that, and given the discussions for the original concept paper and business plan, this concept has led, with a lack of understanding of this concept, has led to a great deal of confusion and uncertainty, both in industry and among regulators. Consequently, the revised version of Q9, 1, seeks to clarify what formality in QRM means. It discusses degrees of formality and the factors that might be considered when determining how much formality to apply to a given QRM activity. As part of this discussion on degrees of formality, it's important to realize that formality in quality risk management is not a binary concept, meaning formal and informal. Given this, varying degrees of formality may be applied during quality risk management activities. So overall, it's fair to say that formality can be considered a continuum or a spectrum from high to low. So when I think of degrees of formality, I immediately think of continuum. Point 3, related to this continuum concept, the third point highlights the revised guidance discusses the characteristics of higher and lower levels of formality. Again, I believe there's a quarter to a half a page on the specific concept within the guideline, and of course there's additional training material on this as well. And then 4, not surprisingly, the fourth point emphasizes there's flexibility in how much formality may be applied in relation to QRM activities. And I think this is probably expected given the fact that there's distinction between higher to lower, given the nature of the continuum, and given the nature of the varying degrees. Flexibility is something that's going to be understood, again, hopefully well developed by the industry and regulatory bodies. But let's take a step back. What is the real benefit of understanding formality in the QRM context? Well, EWG thought about this, and they came up with a few ideas. We felt that additional clarity on formality may help ensure that the extent of scientific and methodological rigor applied during QRM is commensurate with the level of risk. In addition, and this came up a great deal in discussions, was that understanding formality might also lead to a resource savings for QRM and the use of these resources more efficiently. For example, where lower risk issues are dealt with more efficiently via less formal means, the resources from that would be freed up for managing higher risk issues and more complex problems. That's kind of a complicated sentence, but I think it's an understanding that, again, if you understand and apply the right level of formality, the concept is you'll be able to shift resources when higher risk issues arise. And again, before we leave this topic of formality, I want to reiterate the value of reading the ICH guideline in connection with the training material. As you can see over the last few minutes, it's pretty difficult to explain formality, let alone the details of a continuum involving formality, in just a few minutes. So as I mentioned before, there are specific training decks for each of these 4 topics today. Topic 4, risk-based decision-making. While ICH refers to decision-making, there's a lack of clarity in the original version of Q9 in terms of what good risk-based decision-making is, how it might be achieved, and how QRM may improve decision-making generally. I think we can all agree that the COVID-19 pandemic illustrated the importance of effective risk-based decision-making, both by regulatory agencies and the industry. And so given this, the revisions in ICH Q9 and associated training seek 1, of course, to provide clarity, and 2, to address the expected benefits of investing in risk-based decision-making activities. So let's start with the question, where does risk-based decision-making occur? Well, ICH, the revised guideline says that risk-based decision-making is inherent in all quality risk management activities. It provides an essential foundation for decision makers in an organization. The guideline also indicates that the decisions made from quality risk management activities include those in relation to 1, what hazards exist, 2, the risks associated with those hazards, 3, the risk controls required, 4, the acceptability of the residual risk after risk controls, and the communication and review of quality risk management and activities and outputs. Bottom line, effective risk-based decision-making begins when determining the level of effort, formality, and documentation that should be applied during the quality risk management process. For topic 5 is hazard identification. This may seem like a fairly minor, but we believed it was an important change in the guideline. And the term risk identification has been changed in the revised guideline to hazard identification. This was done to better reflect the guidance in the original ICH Q9 document. The figure in ICH Q9 on the left depicting the QRM process was also changed to replace risk identification with hazard identification. And as you can see in the blue box, the change aligns with the expectation to identify hazards relevant to patients when evaluating risks, and it may improve how hazards are perceived and assessed. And topic 6, risk review. The ICH revision work continues to provide additional clarity on the expectations relating to keeping risk assessments current and the implementation of risk reviews. 2 points for this. It's important for this concept to take lifestyle manufacturing performance and quality feedback into account. And 2, risk review ties in the concepts of continuous improvement as expressed in ICH Q10, as well as other lifecycle management guidelines. As mentioned before, and as seen in the yellow box, this area was addressed by developing training materials on this topic only. So in summary, I hope you found the last few minutes helpful to learn a little bit more about ICH Q9. Again, we talked about the purpose, we talked about some milestones and timelines, and we discussed the specific topics. But again, I would be remiss without, as a final reminder, saying, 1, please don't just read the guidelines. Read both the guidelines and the new ICH training material. I'm not sure that reading just the guideline would be classified a hazard in Q9 terms, but I'm confident that all members of the EWG and the IWG would strongly recommend reading both the guideline and the training materials. I look forward to answering any questions you may have for this presentation. Thank you for your time.
Thank you for the great presentations. Our final presentation before the Q&A panel is on the Cell and Gene Therapies Discussion Group by Dr. Kathleen Francison, Global Head of Cell and Gene Therapy Regulatory at Genentech Roche Group. Please join me to welcome Dr. Francison.
It's a pleasure to provide you with an update on the Cell and Gene Therapy Discussion Group. My name is Kathy Francison, and I'm the Global Head of Regulatory for the PT Cell and Gene Therapy Organization at Roche Genentech. I'd like to describe the rather newly formed Cell and Gene Therapy Group, provide the rationale for its formation, outline the scope of our work, and describe the deliverables in the work plan today. The formation of the Cell and Gene Therapy Discussion Group was proposed by the Biotechnology Innovation Organization and was adopted by ICH last August. I serve as the rapporteur of the group representing Bio. I have the privilege of working with Dr. Melanie Eco of FDA CBER. She's the regulatory chair of the group. We held our kickoff meeting last October, and our work plan was endorsed in December. This is a technical discussion forum rather than an expert working group, so our remit and deliverables are a bit different than the expert working groups you've heard about. Rather than revising an ICH guideline, we discuss scientific and technical issues related to ATMP development, and our ultimate deliverable is a strategic roadmap rather than a revised guidance. As mentioned, Bio proposed the formation of the discussion group based on the size and impact of the ATMP field, which has seen tremendous advances in the recent several years. There are now numerous clinical trials that are being conducted increasingly globally. In our database search, there are approximately 1,400 active ATMP clinical trials around the world. And in terms of the product modalities being evaluated, the largest number of clinical trials are with CAR T-cell products, followed by AAV-based gene therapy products. And it was estimated that approximately 5,800 patients have been dosed with AAV gene therapies, and about 38,000 patients have been dosed with CAR T-cell products. This includes both clinical studies and dosing with commercial product. So this is just one indicator of the size and magnitude of the cell and gene therapy field today. As mentioned, ATMP clinical trials are increasingly global. The largest number of studies are being conducted in the US, followed by Asia Pacific, and then Europe. There is also increasingly global commercialization, with an estimated 104 products approved globally, and more products being approved in multiple countries or regions. To date, FDA CBER has approved 27 gene therapy products, including both in vivo and ex vivo gene therapy products. Applying the current ICH guidelines for traditional biologics does not fully address the unique characteristics of ATMPs and can even cause some additional challenges. The development of ATMPs can be uniquely complex across the disciplines, including non-clinical and clinical, and manufacturing and quality development. There are unique circumstances posed during the development of cell and gene therapy products. So it's important now to converge on a science-based regulatory framework across all the regions to make clear the development and commercialization requirements for these innovative products. With the ICH Management Committee's endorsement of forming the discussion group, our overall aim is to develop a strategic framework to address future harmonization needs for ATMPs. The roadmap should include prioritized areas of most need for harmonization where we can reach technical consensus, and it will include specific recommendations for new guidelines to be developed as needed or revisions to existing ICH guidelines. We were also asked to work in close coordination with the IPRP and the World Health Organization to ensure a holistic approach to harmonization efforts and to minimize any duplication of effort. We were also asked to advise existing expert working groups who are working on guidelines where ATMPs are in scope. For example, in December, we were asked to review the ATMP annex for the ICH Q1, Q5C expert working group on stability, and we completed this in January, answering their key questions and doing a detailed line-by-line review of the annex. We anticipate advising more expert working groups going forward as they encounter ATMP topics. Our initial work for the Cell and Gene Therapy Discussion Group is focused on product modalities of rather high maturity, meaning classes of products that have achieved global marketing authorization or have been prominent in global clinical development programs. These essentially include in vivo viral vector-based gene therapy products and ex vivo genetically modified cells, including both autologous and allogeneic products. The work plan reflects the milestones outlined in the REMIT paper, and we have efforts on several milestones and deliverables taking place in parallel. I'll briefly highlight the deliverables here and then say a bit more on the following slides. So our deliverables include a high-level principles document, a global regulatory framework where we level set or build a common understanding of the current framework, identify areas of divergence and harmonization and regulatory expectations, and then do a stepwise review of the existing ICH guidelines for their applicability to ATMPs. Ultimately, we then generate a holistic or a comprehensive ATMP roadmap and roll all of this up together in a recommendation paper. We're currently discussing the maturity level of these products, which are generally not considered to be well characterized, but are considered to be sufficiently mature for harmonization. We have also begun work on the high-level principles document, which you could think of as describing what makes ATMPs different from other pharmaceuticals and biotech products. In addition, in order to build a common understanding of the existing global regulatory framework for ATMPs, we compiled guidelines from across the regions, and we now have a draft global map that contains about 400 documents. This map simply gives us a broad view and hopefully an appreciation for the breadth and complexity of the regulations that need to be navigated. We've reviewed this global map with the regulators in the group and are scheduling interagency meetings amongst the regulators where they can discuss the areas that are already harmonized, identify areas of divergence or gaps, and compile the information and the perspectives that they gain. Meanwhile, we've also begun gathering the industry perspective to see where there is, say, a Y in the road, so to speak. The trade associations are scheduled to meet and align on how best to gather the information, such as a survey of the member companies, where we can then gather the experience that developers encounter while they're conducting global clinical trials or bringing ATMPs to market in multiple regions globally. So the inter-association meeting this month will include Bio, FPA, IFPMA, JPMA, and PhRMA to gather this information. Importantly, we will bring the perspectives of the regulators and the developers together when we collectively discuss the identified areas for harmonization and their priority. This is really a key step in identifying the topics for harmonization, their readiness for this, and their priority based on the collective experience of the discussion group members. Indeed, this group includes many experts with extensive experience with ATMPs, and I'm looking forward to this evaluation. Once this is consolidated, this will all come together to essentially create a lens for our examining the existing ICH guidelines, which will really be quite valuable since actually we will be evaluating all of the ICH guidelines across all the disciplines, including quality, safety, efficacy, and multidisciplinary for their applicability to ATMPs. And according to our page count, there are about 2,000 total pages of ICH guidelines. So having this lens will be very helpful in our doing a strategic assessment. Once we've conducted our assessment of the current guidelines, we will identify areas that most need harmonization and will recommend priorities for revisions to existing ICH guidelines and or new guideline developments as needed. This should then create a staggered approach to be used rather than trying to address all of the needs at once. Finally, we plan to prepare a recommendation paper that will summarize the high-level principles and these prioritized recommendations for revisions to the existing guidelines and or new guidelines. And this deliverable is due in October of next year. Our plan is to submit this to the ICH Management Committee for review and endorsement at that time. So this, in a nutshell, is the ambitions for ATMP regulatory harmonization through ICH. And you can find our remit paper and our endorsed work plan on the ICH website under Reflection Papers tab. You should also be able to find it at the link that I've provided in the slide here. So with that, I'd like to thank you for your attention, and I'm happy to take any questions.
Hello, everyone. My name is Nick Orfanos. I act as the ICH coordinator for Health Canada. I will be facilitating the Q&A session for this meeting, along with Jill Adelberg from the US FDA, who you heard from in the ICH overview presentation at the beginning of the meeting. Before we get started, I just wanted to thank all the speakers today for very informative presentations. Today, you heard presentations on guidelines that have recently reached step 2 of the ICH process. which again, as a reminder, is a draft guideline that was recently finalized for regulatory consultation in the ICH regions, as well as guidelines that have recently reached step 4 of the ICH process, which is essentially guidelines that have recently been finalized at ICH and moving towards implementation in the ICH regions. Today, you really heard a subset of the work of a much larger number of topics being undertaken by ICH, and really shows the magnitude and impact of the work of ICH. So without further ado, we can get right into the Q&A, and I will start by asking questions for Jill, who presented the ICH overview at the beginning of this session. So the first question for Jill, you touched on this briefly during your presentation, but how does ICH determine which new topics are chosen? What is the process, and what are the criteria considered in deciding a new topic?
Thanks very much, Nick. That's a great question. So there are really a number of factors. The first is capacity, or the availability of expert resources to take on a new topic. For example, when the pandemic hit, ICH had a really large existing workload of quality topics and a lineup of priority quality topics that were waiting in the wings. During the public health emergency, our experts acquired added responsibilities that had them struggling to balance overstretched workloads. As a result, the ICH Management Committee and Assembly recommended a temporary halt to the consideration of new quality topics to allow parties to catch up. And in the past few years, that's exactly what the working groups have done. Another factor is that there has to be sufficient scientific understanding of a new topic. All parties may agree that a topic is a high priority, but if there isn't sufficient scientific understanding of the underlying issues, the topic is unlikely to be ripe for consideration. And then I'd mention that in recent years, ICH has advanced new topics strategically by developing reflection papers on emerging topics and outlining potential new guidelines or modifications to existing guidelines via the reflection papers. Often the reflection papers lay out a staged approach involving one or more guidelines. For example, the current revision of the E6 guideline was recommended in the Good Clinical Practice Renovation paper, reflection paper, and the General Considerations for Patient Preference Studies guideline was recommended for consideration in the Patient-Focused Drug Development reflection paper. So really a combination of a bunch of factors, but a great question.
Great, thank you for that, Jill. So next question, what is the process for offering input and ideas for consideration into draft guidelines when they're released for consultation? And some of those which will probably be relevant for some listeners here today.
So all new ICH guidelines and major revisions to existing guidelines are issued for public comment. Individuals and organizations can provide comments through their country or region's formal public comment process or directly to ICH. If you take a look on ICH's website, you'll see a tab for work products and underneath a link to the public consultations page. Currently, the E2D draft guideline is listed along with instructions for submitting comments directly to ICH. All comments are reviewed and assessed by the expert working group developing the guideline and often lead to changes that are reflected in the final guideline. I might also note that E6R3 developed the first stakeholder engagement plan, which recognizes that patients, academic clinical researchers, and others have unique and important insights on the design and conduct of clinical trials, and their voices are relevant and should be heard. And the E6R3 EWG has regularly sought input from these stakeholders. And I think those are the main ways, Nick. Back to you.
Great, thanks for that, Jill. I think we have time for a couple more questions. So next one, I think this is one that we're all sympathetic to, but what has been the impact of the substantial growth in new ICH members and observers over the years?
So it's really been positive. It's contributed to an infusion of new talent and diverse perspectives. And it's also given smaller countries and regions the opportunity to network and learn from more experienced and well-resourced colleagues who are managing many of the same challenges.
Great, thank you, Jill. And one more question. I understand that the regulatory members of ICH meet separately to discuss ICH issues. Can you explain a little bit more about this?
Yeah, another good question. ICH has been tremendously successful because it includes both global regulators and industry experts working together to develop consensus-based guidelines. Regulators obviously have a unique role and perspective, and they do meet regularly under the rubric of the International Pharmaceutical Regulators Program, or IPRP, to exchange information on issues of mutual interest and to enhance collaboration and regulatory convergence. IPRP members typically meet immediately after ICH biannual meetings to discuss implementation of ICH guidelines and other standards, and to share information on global regulatory issues and emerging technologies.
Great, thanks very much for that, Jill. So Jill, I'll pass it off to you for the next set of questions for the next presenter.
So our next set of questions is for Kelly. Kelly, when DDI studies are designed, how are the drugs that could possibly interact with the therapeutic product of interest selected?
That's a great question, because obviously there are endless numbers of drug combinations that could be studied. But so the guideline does address this question. The initial decision to conduct a clinical drug interaction study is based on in vitro study results. So if the in vitro results indicate that a drug might inhibit or induce an enzyme or transporter, or it's a substrate of an enzyme or transporter, the drugs to include in the study are selected based on that mechanism. So the tables in the appendix can help select the drugs. It's preferable to use an index drug if one's available for the specific mechanism. And this is because the results of an index study can be extrapolated to other drug combinations. However, if an index drug is not available for the mechanism, or if the index study has already been conducted, it's typical to select a drug based on mechanism, so there may be an interaction, and the importance of the other drug to the intended patient population. And there may also be risk considerations. Is it considered a risky combination? Thank you.
That's great. That's great. The next question is, are there significant differences between the draft guideline and what we can expect in the final guideline?
Most of the changes were really for clarity. There were some comments that indicated that there might be a misunderstanding of what was in the draft guidelines. So that's what most of the changes were based on. However, there were some changes, and this includes some topics that I included in my presentation. There's more information about when the protein binding measure doesn't need to be capped for a highly protein bound drug. There's more information about the use of endogenous biomarkers to determine the potential for a drug to affect transporters. And there are also some changes to some of the in vitro cutoff values. But other than that, it's mostly clarity related changes.
Great. What issues has the expert working group struggled with most to develop a harmonized approach?
I think the biggest struggle, and this happened during the initial drafting, and then also when we were responding to comments, was really the level of detail and how prescriptive we wanted the recommendations to be. It was interesting that for some of the topics, when we were looking at the public comments, for the exact same topic, we had requests to be more flexible and also to be more prescriptive. And so we had to think about how prescriptive do we really need to be, and when could we just explain a process but not have cutoff values. So that balance was probably the biggest struggle. But there was quite a bit of discussion also around the protein binding topic, the level of detail regarding the use of predictive modeling, because this still is an emerging area. And then there's another ICH guideline related to modeling in general that's really just being started. And then also how to describe the interpretation of drug interaction results, because that can come across as quite subjective. So we had a lot of discussions on that topic.
Great, thank you. One of our participants asked, can drugs that are not listed in the tables in the appendices of the guideline be used in clinical DDI studies?
Yes, that's a good question. Yes, that they can. That is certainly permissible, and it's really expected. Also, there's a preamble before most of the tables in the appendices, and that indicates how the drugs were selected for that particular table. So based on the criteria in that preamble, other drugs may be selected for the drug interaction studies, and then drugs may also be selected based on likelihood of co-administration, the mechanism, and the risk for the patient population.
Great. And our last question for you right now is from a participant who asked, after a DDI study is completed, what criteria can be used to assess the need for a DDI management strategy?
Thanks for the question. And before I answer that, I'll indicate, what a management strategy is. So a management strategy, it can typically be an instruction to not use the drugs together. It could be to change the dose of one or the other drug, or it may be special monitoring that's needed when drugs are combined. So in order to consider the need for this type of strategy, it's important to consider the anticipated change in drug concentrations. So will the concentrations go up? Will they go down? And what is the magnitude? And then based on the knowledge of exposure response and for safety and for efficacy, is there an anticipated consequence of the drug interaction? So that's the most basic way to explain it. And that is outlined in the guideline. Thank you.
Very helpful. Thanks, Kelly. Nick, back to you.
Great, thanks for that, Jill, and Kelly as well. So our next set of questions will be for E2DR1 post-approval safety data management, definitions and standards for management and reporting individual case safety reports. Our presenter was Craig Zinderman. So just questions here. So first question is, are there plans to integrate IDMP into adverse event reporting?
Yeah, so thanks for the question. E2D is really a guideline that's at a higher level, what some might call a foundational guideline. It defines terms, concepts, reporting requirements, and approaches. Guidance on a specific taxonomy like IDMP for product identification would be covered in the E2B guideline. It's really outside the scope of E2D, and it wasn't part of the E2D working group's deliberations. That said, there is an FDA guidance for industry called Identification of Medicinal Products, Implementation and Use, and that's from March 2023. And that guidance describes FDA's position and current thinking on IDMP and the 5 IDMP standards and progress so far with implementing them. And it mentions in that guidance sort of a phased approach where FDA is planning to work with stakeholders to globally implement these standards.
Great, thank you for that, Craig. Next question. The new draft E2D guideline says that a username or a handle isn't enough to confirm the existence of a real patient when cases are submitted via digital platforms such as social media, but that the addition of an identifier such as gender, age, or age category would be enough. In a world where fake profiles are increasingly prevalent on social media sites, could you speak about the expert working group's thought process for verifying the existence of real patients on digital platforms?
Yeah, certainly a timely question. While it was a major purpose of the guideline to come together to address the burden and volume of all of this post-market safety data and post-market reporting from a variety of new sources, including digital platforms or internet, social media sources was one of them. It was really about addressing the volume and how to manage all of these cases and how they should be reported. Specifically, being able to discern the veracity of information on social media or other internet sites, being able to tell what's real and what's not real, what's fabricated by a bot or generative AI, wasn't really part of the goal of coming together to do this revision. That said, it was a big topic for the working group, particularly since many of our discussions happened during the time of, recently with excitement and expansion of AI, really globally. So we provided the criteria that you mentioned for the username to not in and of itself qualify as for patient identifiability, but something else like gender or age would be required to really know the existence of a real patient. So that was sort of a baseline that we were able to provide as a recommendation in the guideline, but really couldn't find other recommendations that we could give that would help, or that would really work to tell what's real and what's not real out there on the internet. That was pretty much what was doable as a recommendation.
Great, thank you for that, Craig. And then one last question, very simply put, what's new and different about the revised guideline?
So I addressed a lot of the differences in my talk and focused on them, but I'll pick two that I think are really important to point out. One of them is that the previous guideline, the original guideline, is really about expedited reports only, whereas this guideline really expands the scope to all ICSRs. Part of the reason for that is the original guideline. I think it really directed expedited cases because there was harmonization with the requirements across regions about what was required, what the time frame was, what an expedited report, what was considered to be an expedited report, serious and unexpected. But now that has changed, and there's not as much harmonization even among what's considered an expedited report. So there wasn't a reason, it wasn't really an advantage to just restricting the guideline to expedited reports. And the concepts that we talk about, digital platforms, patient support programs, definition of an AE, serious AE, really apply to all ICSRs, not just expedited. So we really wanted to broaden it. And the other thing I'll point out is all the changes relative to these new or more frequently used data sources. So for patient support programs, for digital platforms, there are definitions for these things that are provided. These are new definitions. Particularly for patient support programs, the definition really requires that the program has two-way interaction. So that considering adverse events from those programs to be solicited really means that those adverse events are derived in the context of that interaction between the company and a healthcare provider or a patient.
Great, thank you very much for those responses, Craig. So Jill, back to you for the next presenter.
Terrific, thanks very much, Nick. So Dr. Legaré is unable to join us this afternoon. In her place, we're delighted to welcome Dr. Keire Alzarad. Keire is the E6R3 rapporteur and has expertly led the working group during development of the revised principles guideline, Annex 1 and now Annex 2. In his spare time, Keire serves as the director of CDER's Office of Medical Policy, where he leads the development, coordination, and implementation of medical policy programs and strategic initiatives. So Keire, thank you very much for joining us. Our first question is, why doesn't ICH E6R3 apply to non-interventional trials?
Thanks so much, Jill. Do you hear me?
Yes, yes.
Oh, okay, good. Perfect. And thank you again so much, and I'm delighted to be here with all of you. It's a great question. Actually, the EWG, the expert working group, from the very beginning discussed the inclusion of non-interventional trials, what's called actually non-interventional studies. It's important to note that having an intervention is really a hallmark of clinical trials. Obviously, non-interventional studies, or as a term called like observational studies, do not have an intervention. And if you think of E6, it's really an outline of how to manage the risks, but also how to manage and ensure the quality of the data, the reliability of the results. In the context of having an intervention, that's very different than when you would not have an intervention. For example, observational studies. For observational studies, the risk profile in general is more focused on data privacy and confidentiality because of the lack of the intervention. So we thought that's really a very different topic to address. Not that's not important. It is important, but for E6, we want the focus to be on interventional studies, hence clinical trials, and that will give us an ability to provide more of a concise and targeted and reasonable approach across the board, across the different designs of clinical trials. I hope that's helpful.
That's very helpful. Thank you, Keire. Our next question is from a a participant who said, I may have missed this, but is ICH E6R3 currently in force? If not, what's the anticipated timeline for implementation?
Yeah, thank you so much for the question. We are on time to hopefully finalize R3 by the end of the year. Right now, R2 is finalized, and it's important to note that there is no contradictions between R2 and R3. R3, we've seen it as more of a modernization step that took the concepts in R2 and really put them in a more of a modernized context, if you would, with proportionality and risk-based approaches at the core of all of that. I hope that's helpful.
Great, thank you, Keire. Next question is, the E6R3 principles are very broad. Do you plan to develop training materials, including examples, to help sponsors better understand some of the concepts discussed?
Yes, great question and very timely one. As we developed E6, we realized that there is a lot of areas that worthy of a little bit deeper dive, if you would. And we made a list. This list now is a couple of years old and it keeps accumulating. We made a list of the key aspects that we would like to address in the training. We're hoping this training will not just be a repetition of what's in the guidance, but it will have use cases, for example, novel trial designs, trials with decentralized settings, trials in healthcare, embedded in healthcare, and to show through that training itself how GCP can be flexible and agile in different contexts here. So yes, the training is a primary aspect of what we would like to do, and we're putting quite a bit of onuses and hope on the training to complement the guideline itself.
That's great. The next question is, were all public responses and comments provided on E6R3 reviewed? How were the numerous comments taken into consideration for updates? And finally, were the comments reviewed during the series of joint meetings?
Yeah, it's a good question. We've received around 6,000 comments during the public consultation period. We were really excited to see the level of interest in E6. And we actually very much committed to really looking at every single comment. What we did, each agency looked at the comment that we received at the specific regions. Like for example, for FDA, we looked at everything collected from the US. And what we had to do is to put those comments into themes. The group came up with, I think, approximately 17 or 18 themes that the totality of the comments really fits within those themes, with a few exceptions. Obviously, we have a miscellaneous category. And now the group, through their meetings, they're going, with on each theme and addressing each element here. It's been really helpful for us to see where the concentration of the comments really try to point us to. And we believe in general, this is going to make the guideline far more robust.
That's great, Keire. So in the interest of giving every presenter an opportunity to answer questions that we've received, let's move on now to go back to Nick and move to M14. And if time permits, we'll come back for a second round of questions. Thanks very much, Keire.
Thank you.
All right, thanks everyone. So yes, our next presenter was on M14, which is General Principles on Plan, Design, and Analysis of Pharmacoepidemiological Studies that utilize real-world data for the safety assessment of medicines. So Ken from Health Canada was our presenter here. So one question for Melissa. A reflection paper on harmonization of real-world evidence terminology and convergence was released last June for public consultation. How does the topic of that paper relate to or complement this M14 guideline?
Thanks so much for the question, Nick. And it's a really good question. So the draft reflection paper, and it's available on the ICH website for those of you who may not have had a chance to look at it during the public consultation last year. It's really meant to address some of the broader challenges that we have related to harmonization and convergence that we have both in the real world evidence and real world data spaces, with more of a focus on effectiveness, whereas M14 is more specifically targeted to the safety assessment of medicine. So just as a bit of history for the reflection paper, it really came out of themes that were identified by The International Coalition of Medicines Regulatory Authorities, RWE workshop participants back in June of 2022. So this led to a resultant ICMR statement on international collaboration in the RWED spaces that was released in June of that year. So these were really kind of the ideas of a group of collective regulators trying to address this. So I will say that really in the end, both are meant to complement each other. So both the topic of the reflection paper as well as M14, in the sense that they both focus on promoting the use of high quality data and evidence, and better enabling the integration of both of these into regulatory decision making. So as we have worked on both M14 and the draft reflection paper, we have, of course, been very mindful of avoiding duplication and inconsistencies amongst the two. So, for the reflection paper, it's really outlining objectives, promoting more of the common understanding of, different types and scopes of real-world data and evidence, description, ultimately really the goal of informing assessment for regulatory purposes, while also looking at some different areas. So for example, common principles related to things such as protocol and report formats, and promoting study transparency. So again, I think they're both complementary to each other. And we really do welcome comments as the reflection paper does progress through the process, and should a topic be proposed, please keep a lookout for that. So thank you.
Great, thank you for that answer, Melissa. Jill, back to you for the next presenter.
Terrific, thank you, Nick. And hold for 1 second, our live audience. So I'm going to ask a few questions on Q2R2 and Q14 for Dr. Keire. Dr. Keire, did the Q2, Q14 EWG consider the changes being proposed in the M4Q Common Technical Guideline guidance? Excuse me.
Thank you for the question. Yeah, we actually worked really closely with the folks that were working on M4Q, which is, of course, kind of early in the process compared to where we were. So we're kind of giving them input on elements that we were working on. So there was a lot of discussion, and ultimately we decided, because there's uncertainty in actually what M4Q is going to contain, that we wanted our guidelines that were going to be implemented much sooner to be functional guidelines, in the current state. So we included elements of the currently enforced M4Q CTD guidelines in our submission section. But certainly those will need to be updated when M4Q is modified. Thank you for the question.
Thanks very much, David. One of our participants asked, as demonstrated in your slide by the dotted line, currently you have to flip between ICH Q2 and 14, yet we are promoting the lifecycle approach. Keeping ICH Q2 as a separate document, unfortunately, still gives the perception of validation, being a separate activity, when in fact it is integrated with development and continued verification of the true analytical procedure lifecycle. What was the reason behind separating Q2, R2, and Q14? And are there plans to combine the documents?
Yeah, thank you for the question. And I guess I'll need to take a minute here to kind of take it apart. I mean, I think an analytical procedure has a life cycle. I don't think we're promoting that or otherwise, speaking to an analytical life cycle. And so, but I also would say that the EWG, starting back in 2018 in Charlotte, I still remember this conversation about whether we should have one document or two. And a lot of the discussion, I would agree with the question that, what is developed is validated, right? And but then when you think about how these documents, these guidelines are used, I mean, some folks may not want to use the tools that are available and the kind of enhanced approaches that are described in Q14, but they all will use, Q2 elements. right? And so if we combine them into one document, those key validation elements are diluted in this other document, and so ultimately it was decided to keep two separate guidelines, but that was after a lot of discussion. I would say that discussion went on for 3 years, and involved a lot of the constituent review. So it wasn't done idly, I guess. It was, and I think that, but our focus was on the the end use of the guidelines. Some will just use Q2 R2, while others will take advantage of what's presented in Q14. So thank you for the excellent question.
Great. Our next question is, when does FDA expect to implement Q2 R2 and Q14?
Well, hopefully in the next few months. So we've been going through all the steps, and since November, to get to the implementation phase. So look for it soon. Hopefully it will be appreciated when it's available. Thank you.
Great, thanks, David. The next question is, what are the benefits of using an enhanced approach to analytical procedure development for applicants?
Right. So there's a couple parts of this. One is I think a lot of the firms do in development, they learn a lot about their analytical procedures, and then they kind of put it on the shelf, do a validation, and submit that to a regulatory agency. And really, sometimes, as I mentioned in the presentation, sometimes the regulators don't fully understand, maybe something about a particular non-conventional technology, and so that leads to a lot of questions and delay in time. And so the idea is that we can avoid that by front-loading this work, right? And so you would apply an enhanced approach to your in analytical procedure development, develop a lot of procedure information. Maybe you're already doing that kind of thing, and this kind of gives you a tool to present it to the regulator and say, hey, look, I really have this under control within this operation space. I know that it's going to give me good results, and the regulator could say, I agree with you. That you can move around in that space without really having to do another filing, right? And so that's kind of the idea. That kind of pulls in Q12 tools, but again, ultimately, the enhanced approach gives the applicant a lot more procedure knowledge that can avoid problems, right? So even if you don't do anything and submit it to the regulator, it's still going to help you. That's my point of view. So thank you.
Great. The last question for you, David, before we move to the next presenter is, how do the Q2, R2, and Q14 guidelines relate to the guideline on continuous manufacturing?
Yeah, good question. Thank you for that. So in continuous manufacturing, there's a need to carefully control a process, and that is done by using process analytical technology tools. Often they're multivariate spectroscopy tools where you're collecting the entire spectrum rather than an isolated wavelength, and you're using a model to provide real-time feedback to a process to keep it under control and produce drugs that are the same quality over that continuous process. And so it's the kind of things that we provide in both Q2 and Q14, I think, are going to be tremendously useful to folks that are working in that space. And because we've expanded the descriptions to account for those kinds of that application. That was a real target in our discussions. And so I think it's really going to be very helpful to folks working in the continuous manufacturing space. Thank you.
Terrific. Thank you, David. Nick, back to you.
Great, thank you, Jill and David. So the next set of questions is for our Q5AR2, which is viral safety evaluation of biotechnology products derived from cell lines of human or animal origin. So our presenter here was Christopher Storbeck from Health Canada. So Chris, our first question is, if head-to-head testing is not required for NGS validation, what is recommended for NGS validation when used for cell bank testing?
Hi, Nick. Thank you for the question. So for validation, excuse me, guidance is provided regarding validation of the method, and performing matrix-specific validation when NGS is used as a replacement method is recommended, and qualification of the method in matrix-specific verification if NGS is used as a supplementary method. So it's also clearly stated in the guidance and the revision that the validation and qualification should consider the principles of ICH Q2. Thanks.
Thanks for that response, Chris. So the next question is, are cell therapies considered to be within the scope of the ICH Q5A revision?
So thank you for the question. Cell therapies are actually excluded from the scope of the revision. However, it is stated in the revision that elements of the guidance, such as biological starting or raw material safety considerations, can be taken into consideration. So, of course, cell therapies are not a therapy that's amenable to virus clearance, and thus the reason for its exclusion. But that sort of brings into prominence the other two pillars of virus safety, and they take on a more prominent place in the overall virus safety considerations. So, additionally, the viruses utilized in the generation of some cell therapies, such as CAR T cell therapies, may fall within scope, but that depends on how amenable those viruses are to virus clearance. Thank you.
Great, thank you, Chris. Next question. So whether ICH Q5AR2 is applicable for the drugs that are produced using animals' milk source and shell and beeswax source materials as bulk raw material. Please clarify if this guideline is applicable for the above said animal source materials, non-animal source material that is non-living in nature, not like a cell or biological process-based material.
Well, thank you for the question. So ICH Q5A deals with in terms of the scope, deals with biotherapeutics manufactured from cell lines. So those types of starting materials would fall outside of the scope. However, that's not to say that some of the concepts of ICH Q5A, including raw material sourcing and testing, could not be applied to the viral safety considerations for those types of raw or starting materials. Thank you.
Great. Another question. Next generation sequencing is mentioned in the revision as a method that can be used to replace the in vivo virus detection methods and supplement or replace the in vitro virus detection method. Why does the revised guidance promote the use of NGS over the in vivo methods and in vitro method?
Yeah, excellent question. It's perhaps not so much that it's promoted necessarily, but it's based on the scientific advances and the science behind it, next generation sequencing, and essentially it comes down to the sensitivity of the methods. NGS is a highly sensitive method capable of detecting very low levels of sequences within a mixture. And considering the other methods for detection of viruses, like the in vivo and the in vitro method, aside from reducing the use of animals, the in vivo methodologies, there can be issues with sensitivity, and with the in vitro methodologies, often there are situations where the same cell line may not be able to detect all family members or all members of a family of viruses. So there are definitely issues with detectability and sensitivity with the in vitro and in vivo methods. So that is the capabilities and sensitivities of NGS allow a sort of a very broad general detection capability. And so, of course, if sequences are detected, it has to be confirmed whether these sequences are accompanied by infectious virus. And so it's really a first signal. And in that regard, it is a more stringent form of screening in a sense. Thank you.
Great, thank you, Chris. So I think we can pass it back over to Jill for the next set of questions for the next presenter.
Great, thanks very much, Nick. So the next set of questions is for Steve Mahoney on Q9R1. Steve, how does Q9R1 help industry and regulators better manage the dynamic risks that occur during the product and facility life cycles? Why don't we move on? Nick, why don't we move on and come back to Steve? Should we move on to our next presenter?
Sure, we can definitely do that. Might be some technical difficulties here. Yeah, no problem. So the next set of questions will be for the presentation on cell and gene therapies discussion group, and this will be for Kathy Francison. So first question for Kathy, we noticed that you use the term ATMP during your talk. Is ATMP used interchangeably with CGT product?
Yeah, thanks for the question. It's really a good one. And in fact, what we see is that there are many different terms used to describe this class of products around the world. So the discussion group did really look at this more closely. And as I mentioned in my talk, we've been asked to work closely with the World Health Organization. We do have a WHO rep on the discussion group. So we looked at this question of terminology. And actually, the WHO went through quite an extensive and thorough process of looking at how you delineate these products, be they cell and gene therapies or advanced therapy medicinal product. Those two terms do tend to be used interchangeably. But the WHO did go through and engage global stakeholders in a very transparent manner. And the result of their thorough work culminated in the Annex 3 paper on human cells and tissues and ATMPs, and that was adopted in 2023. And so that's actually a very good resource where you can just see what's considered in scope and what's not, and what is some broadly recognized terminology. And so we actually agreed that ATMP really is the appropriate term to use, with it being already internationally recognized. Thank you.
Great, thank you for that, Kathy. Next question. Are there ICH expert working groups or new topics, I guess that would be, that you would anticipate the cell and gene therapy discussion group to advise of this year?
Yeah, thanks. That's another great question. As I mentioned, we have been asked by the ICH Q1, Q5C expert working group to provide input, and we've done that. We know that the ICH Q6 expert group is getting started this year, working on specifications, and we anticipate that they may reach out to us. Also, ICH M4Q on the Common Technical Document, they are quite active right now as well. And we can imagine that they might also contact us. In fact, with cell and gene therapy products, we do have some very new technologies new types of manufacturing processes, new types of information. And so we can imagine that we would have some pretty unusual dossier structures that would work the best. So that's another one we anticipate. Thanks.
Great, thank you for that, Kathy. Next question, what are some of the major challenges that you have experienced or foresee in completing the remit of this DG?
Yeah, it's an excellent question. It's actually quite a tall order for us to assess all of the ICH guidelines for their applicability to cell and gene therapy products or ATMPs. Actually, this field is more interdisciplinary in many regards. There are many examples where clinical and quality colleagues need to work together closely. So it's actually quite good that we have clinical, non-clinical, and quality experts working on this together. This being said, it's nevertheless quite a large undertaking. We were, I would say, reasonably impressed when we pulled together all the documents that we could and found ourselves with a little over 400 guidelines available today. So we'll be going through to filter those and look to see where there's already some existing harmonization or where there are gaps. So that just showed us that it's going to be valuable to do these initial kind of exercises of level setting before we dive fully into all the ICH guidelines. Thank you for the question.
Great, Kathy. And another question, you may have already answered this, but how large is the DG and how is the DG able to handle consensus on global regulatory differences for cell and gene therapy products?
Yeah, it's actually, this discussion group is quite large. We have 48 members. We have regulators from all around the globe, all different regions. As I mentioned, we also do have the WHO and the IPRP represented here. So consensus really requires some thoughtful discussions, sharing information and documents that are currently in place. And sometimes we find that the intent might actually be aligned fairly well, even though they might be explained using different terms. So this will require some really thoughtful work for us to reach consensus so broadly. Thank you.
All right, thank you very much for those responses, Kathy. So we are going to try to go back to Q9R1 with Steve Mahoney. So let's give this another shot. Back to you, Jill.
Terrific. Thank you very much, Nick. And hold for 1 second. So Steve, how does Q9R1 help industry and regulators better manage the dynamic risks that occur during the product and facility life cycles? So it sounds like we may still have some technical difficulties. So maybe we should go to round 2. And hopefully we can work through these relatively quickly. So sorry about that.
All right, thank you, Jill. So we're actually going to cycle right back to you to go through another quick round of questions for the presenters. So next question is, have there been efforts to improve the M10 bioanalytical guidance as it applies for biomarkers? We have used the endogenous analytes section to adapt our procedures, but in most or all cases, we cannot follow guidance because we don't have a true blank matrix.
So that's a great question. ICH M10 is intended for the method validation of assays for small molecule drugs and biologics. Biomarkers are specifically out of scope. If you wish to follow the principles of M10 for your biomarker method validation, not all aspects of the guidance will address biomarker method validation, and you should provide justifications for your choices in addressing areas that differ from small molecule drug and biologic method validation? Great question. Back to you, Nick. Thank you.
Great, thanks. So I think that concludes the questions we got for you, Jill. So we can actually pass it right back to you for the next set of questions for M12.
So thank you very much, Nick. And it looks like we don't have further questions at the moment on M12. So perhaps we can move to our next presenter.
Yeah, we can move to E2D. We do have one more question for E2DR1 for Craig. So the question is, the definition of D0 for ICSR coming from the literature is different from the position of Inspector of Europe. Do you plan to align the position?
Yeah, thanks for the question. What I can say is that the definition of day zero and exactly how we characterize that was considered very carefully for all the sections of the guideline where we provided a time frame for reporting, but especially for the literature section because we had a lot of discussion about that. So it's absolutely intentional where we landed and the recommendation that we're providing, and all parties, including my colleagues with the European Commission and the EMA, are well aware and understand this definition of day zero and are fully aligned with it. I can't speak to European inspectors or what may or may not need to happen for alignment on their side, but I can tell you it's very intentional, and every group on the EWG is in agreement with it. Thank you.
Great, thank you for that, Craig. So Jill, I think we're passing it right back to you. We're going to give Stephen here another shot with Q9.
Terrific. Thank you very much, Nick. And I'm just going to go back to our list of questions on Q9R1. Bear with me for just 1 second. So Steve, how does Q9R1 help industry and regulators better manage the dynamic risks that occur during the product and facility life cycles? Okay. We're still trying, everyone, but it looks like we're still experiencing some technical difficulties with Steve's mic. So let's go back to E6R3 and some more questions for Keire. Keire, one of our participants asked if you could provide a little bit more information about plans for Annex 2.
Yes, absolutely. As all of you are aware now, Annex 2 touches on design such as the use of decentralized elements, pragmatic elements, and the use of real-world data, specifically in the context of interventional clinical trials. The outline of the guideline is very much in advanced shape at this point. Actually, we started what we call caucus review, which was we sent it to experts within each organization. We included also academic experts in this review. So, and we start getting back their input at this point. And I'm happy to report that so far we've been getting very positive reviews of the guideline of Annex 2 specifically. And the group is working on it diligently to incorporate the input that we've received. The guideline itself is high level, and as Carol earlier in her presentation mentioned, it doesn't necessarily add new requirements, but clarifies how GCB concepts can be applied to those designs. We still anticipate to be on time, which means public consultations in the second half of this year. So hopefully everybody will take a look at it and provide us with input. We're excited to get interest similar to what we had in Annex 1.
Great, thank you, Keire. Next question is, what does E6R3 have to say about patient engagement?
Yeah, it's a great question. We've discussed that quite a bit because in ESEX we are trying to cover the spectrum of clinical trials, all the way from early phase kind of proof of concept when you have very few, sometimes it can be counted in one hand, number of participants, to the more complex, phase 3s, large clinical trials, as you can imagine. So, for early phase, certain level of engagement may not be needed or may not make sense when you have just a proof of concept study in a few individuals, for example. However, not just participants' engagement, but also community engagement may be very relevant at large-scale clinical trials. So the current language encourages engagement with the stakeholders, and they include patients and participants in that, but as well as healthcare providers. For example, if you want to work with community healthcare providers, you might want to consider also engagement with them at earlier stage. And just to be very transparent with everybody, in a lot of the comments we received asking us to bolster a little bit the language we have around community and patient engagement, and that's something the EWG is working on at this point.
That's great. Thanks very much, Keire. Nick, why don't we move on to the next presenter?
Sure, thanks, Jill. Just working through the questions here. So yes, the next presenter will be back to David on Q2R2, Q14. So the question is, how important is it to include a criticality score accounting for impact and uncertainty in risk assessment for analytical methods? And how can this criticality score be integrated into the risk assessment ranking system effectively?
Well, thank you for the question. I think Steve would probably opine on this. Definitely, I'd love to hear actually his answer to this question. Unfortunately, we're having those technical difficulties. But I say that's this criticality accounting is a tool that can be used. We talk about risk assessment in Q14, but we're not advocating the use of one tool over another. That it's good that you're doing it, and that's one approach. And the second part of this question, can it be done effectively? Right. That's kind of an implementation question, and maybe more appropriate for Steve Mahoney to answer than for me. But I think it's a tool, but it's not the only approach that you can take to do these risk assessments. It's certainly one that a lot of people do use, and there's a host of scientific literature describing effective ways of doing that. Thank you.
Great, thanks very much, David. So I think that concludes the questions for Q2R2, Q14. So Jill, back to you.
Great, Nick. So we can move on to some additional questions for Chris Storbeck and Q5R2. Chris, you mentioned that for certain well-characterized cell substrates, in vivo testing may not be necessary during cell line qualification. Could you expand on this a bit?
Yeah, thank you for the question. Certainly, and this pertains to a situation where the sponsor has extensive experience with that particular cell line. It's a well-characterized cell line, such as a CHO cell line, for example. So if they've performed in vivo testing on the parental cell line, and they've generated several master cell banks for other similar products by a well-controlled process, then in that situation, the in vivo testing requirement can be waived. Thank you.
Great. The next question is, could you please give a few examples of the use of prior knowledge to achieve regulatory relief?
Yeah, certainly. So prior knowledge, and certainly the answer I just gave is one example of the use of prior knowledge. There's other examples as well. For example, with the resin reuse, as I mentioned, with the protein A chromatography steps, and also the anion exchange and cation exchange chromatography steps, where in certain situations prior knowledge can be used. regarding reuse of those columns. So, and that comes down to a knowledge of the process, knowledge of the process intermediates. Also for leveraging virus clearance studies for a new product, using data from a well-characterized platform process can be acceptable. So for certain virus clearance steps, such as low pH, detergent solvent inactivation, and virus filtration. In the case of virus filtration, one product-specific demonstration of clearance has to be submitted. That's a recommendation in the guidance. But certainly where the process parameters are well understood, the process intermediates and the composition of those intermediates are understood, then those that information can be used to gain some regulatory relief. But it has to be laid out and scientifically based during the submission. Thank you.
Thanks very much, Gus. The next question is, there were many for your section for Q5A. Suppose a research protocol proposes using a novel biotechnology product derived from cell lines predominantly representing a single ethnic group. The product is intended for a disease that disproportionately affects a different population. Dr. Storbeck, how might the Q5R2 guidance inform an IBC's review of a protocol like the one described? Are there specific concerns the IBC should raise regarding the potential impact of source cell line diversity on the product's safety and efficacy across the intended patient population?
Well, thank you for the question. It is so. It is ICH Q5A does address the manufacture of biotechnological products derived from cell lines, and I think it's more important to consider the species of cell lines with respect to safety, efficacy, and risk, rather than specific ethnic groups within a species. So through the lens of scientific understanding and genetics, and there's certainly going to be some genetic differences, but overall from a species perspective, where the similarity is to such an extent that the granularity of different considerations between ethnic groups, I don't think is possible. But thank you for the question.
Thanks very much, Gus. Okay, Nick, I'm going to send it back to you.
Great, thank you, buddy.
Yeah, sure.
Yeah, sorry. I think we've got a few more minutes left, maybe a chance for another couple of questions. But thank you very much, Jill and Chris, for that. So I'm going to go back to the cell and gene therapies discussion group, Kathy. So the next question is, what are your thoughts on the advancement of the therapeutic products under the initial scope of the discussion group, and how the remit will help in advancement of these products and other cell and gene therapies products, especially in light of the fast speed of technological development of ATMPs?
Yeah, thanks so much for the question. We continue to see quite a number of AAV-based gene therapy products and CAR T-cell products going to market. And they're going more globally. And so that experience helps everyone see where they've really matured and what the requirements are to make it through development and on to approval. The other thing it does is it highlights the current state of where these products are. And so it's helping us think about what are the requirements currently, but what do we think will be required in the future? And that should really help set the stage for anticipating a lot of emerging technologies that are coming along in this field. They do, in fact, come along very quickly. So our working through these initial product classes will be quite helpful. Thank you.
Great, thanks, Kathy. And I think we've got time for one more. So another one for you, Kathy. The discussion group is for cell and gene therapy, but the presentation refers to the term ATMP. Can you please explain the significance of the use of the term ATMP versus cell and gene therapy?
Yeah, thanks, Nick. I think we kind of had that question earlier in it, and you can use either of these terms interchangeably. But indeed, as we looked across for really the best resource, we turned to the WHO and the terminology that they put forward in a very thorough way. It's an excellent resource, and so we do feel that using the term ATMP is quite appropriate. Thank you.
Great, thanks very much, Kathy, for those responses. So just looking towards our coordinators here, or Jill, or do we have time to take any other questions, or are we looking to close off?
I think we're right at time, Nick. Our participants have had great questions. So
great, yeah, no, definitely a lot of great questions, a lot of great information in today's session. Unfortunately, never enough time to get through everything, but we wanted to thank everyone for participating today. We hope you found it very informative. Thank all the presenters as well for their time in providing this very valuable information, and wish you all a great day. Thank you very much.
Thanks so much. We do have a few housekeeping reminders. Please remember the 14-day deadline. That's March the 7th to claim the certificate of attendance to support CE for SOCA, RAPS, SQA, and ACRP. Remember to download the certificate immediately upon creating it. Otherwise, this opportunity will be lost. Recordings will be posted in approximately 7 to 10 business days. On behalf of CDER Small Business and Industry Assistance, we hope that you enjoyed today's presentations and found them helpful. We look forward to your presence at our next SBIA webinar. Thank you for attending, and this concludes today's webinar.